These are in listen-only mode.
Hi, we're going to get started in about 2 minutes here, just to make sure everyone is online, and we'll talk to you then. Thank you. Hi, my name is Brian Haynes. I work for Fluxion Biosciences on the BioFlux project, and it's my pleasure today to introduce Dr. Mark Weber. Dr. Weber started his science education at the University of Birmingham, and then went on to finish a master's degree from the University of Birmingham and to finish his PhD as well from the University of Birmingham on mechanisms of antibiotic resistance. Dr. Weber has continued this work into his postdoctoral fellowship and now as a group leader at the Quadram Institute. Recently this year, Dr. Weber published some fascinating work on triclosan, which was, if you remember from a few years ago, was in everything from acne creams to wound dressings to typical hand soap. And turned out that last year, the U.S. Food and Drug Administration issued a ban that took effect this year on triclosan in most materials. I think Colgate was actually spared from that list, but everything else has it. So now instead we have, I just got some hand soap from our fitness instructor, and perhaps the coconut oil in there, along with the actual SDS detergent, is probably the majority of the antimicrobial properties of that anyway. It's my pleasure to turn this over to Dr. Weber.
Okay, Brian, thank you. That's very kind, and thanks to you and to Fluxion for the invite to talk about some of the stuff we've done with the BioFlux. So first off, I guess hopefully everyone can hear me okay. So what I thought I'd do is just give a little overview of what our group does, our interests, and then give you some examples where we've used the BioFlux in our research. So we're really interested in studying how bacteria respond to stress, and we do a lot of work with biofilms, and the BioFlux has been one of the tools that we've used there. So the structure of the talk, if you like, is I'll just kind of briefly explain what we do, talk a little bit about the BioFlux just to introduce it if anyone's never used one or is interested in maybe using them, but not a huge amount of, I'm not going to do kind of big protocol details at any point in this. And then we're going to just talk about a few different areas where we've used it to kind of compare biofilm formation between isolates, to look at some anti-biofilm therapies on, or potential therapies on their ability to inhibit biofilm formation, and then to look at what's happening within populations. So we've done quite a bit of single cell stuff looking at viability in biofilms, gene expression, and then also we've done a little bit of playing with using it as a tool for kind of doing evolutionary studies. So I'm just going to cover the principle behind each of these areas and kind of some brief examples. So that's the kind of plan, and I'm not quite sure how long this will be, but not too long, so don't worry, we're not going to go on forever. So what do we do and who are we? So my kind of potted history, so Brian's just kind of alluded to, so I'm actually from London originally, so these three skylines give the kind of life history. So lots of people think I'm from Birmingham because, as Brian said, I went to Birmingham, did a degree, a master's, a PhD, bit of postdoc, got a fellowship, started a group. So I got well and truly stuck in Birmingham, which is the second biggest city in the UK, if anyone doesn't realise. And then moved just over a year ago now to Norwich. So Norwich is very different as an old medieval city, whereas Birmingham was an industrial revolution city. But what I've been doing, what I'm interested in is really understanding how do bugs become resistant to drugs. So we're interested in antibiotic resistance, and really we try and understand how, when, where, and why bugs become resistant to drugs, with the ultimate goal of trying to use some of that knowledge to use our drugs better or come up with new ways to prevent infection as our ability to treat infections becoming challenged. So the kind of main interest of the group is we have interest in some specific antibiotics. So we're interested particularly in quinolone antibiotics a lot. We're also interested in multi-resistance, so how bugs can become resistant to lots of different classes of drugs in one go. We'll talk a little bit later about something called efflux, which is where bugs can produce pumps to throw out antibiotics and other things. We've done a lot of work looking at biocides, antiseptics, and how bugs can evolve resistance to those. And then a lot of that links into biofilms. So obviously bacteria in the real world, most of them are in a biofilm, which are bugs adhered to a surface of some kind or to each other in aggregates. And biofilms are very, very hard to kill. And then so we're interested in how do bugs, how can you kill biofilms? How do bugs in biofilms respond? We're also interested in kind of plasmids and gene transfer and other species. And we work with lots of species. So we work with Salmonella, E. coli, Acinetobacter, Staph. I do know how to spell aureus. That's an autocorrect from PowerPoint. Apologies. Pseudomonas, Klebsiella, Campylobacter, and others. So we work with a lot of organisms, but the principles are the same. So just one more kind of brief slide about what we do and how we do it is we really kind of like to think of we have two laboratories, the real world being one, so looking at isolates that have come out of patients or animals or contaminating an industrial process to try and see things that have come from the real world. And then also we do a lot of experimental evolution, so we look at controlled conditions in the laboratory. So we do these kind of experiments, we do a lot of phenotyping, biofilm assays, looking at when things die and how they die. And then we do lots of characterization of isolates, and so we do lots of sequencing or kind of massive mutant libraries, transcriptomics, proteomics, etc., etc. And increasingly, we've been interested at the responses of individual cells within populations rather than whole populations. So confocal microscopy and flow cytometry we've used quite a lot, and the BioFlux is a very nice platform for presenting samples for confocal, which we'll cover. Okay, so that's a very brief history of what we do. So we're really interested in how bugs become resistant to drugs. Okay, so the BioFlux. So what is it? So I guess some of you probably maybe have one, some of you may be thinking about have one, some of you may not. So just for those that are naive, the BioFlux is kind of looks like this. So it just looks like a kind of box. And the box is attached to a special plate. And if you look at this top left-hand part of this panel, then you can see the box connected to the plate by the kind of airflow. And the plate on the top right, you can see that you have these matched wells. So this is an example of a 48 well plate. There are various other standards that you can get. And then you have an inlet well and an outlet well, and they're matched by the microfluidic channel that's in between. So this is the kind of the magic part of the BioFlux plate, is that you have these two channels, and then linking them is this small microfluidic. And then the bottom left, you see a kind of a zoom in of that microfluidic. So basically you have your inlet well and your outlet well, and they are linked. And you can control the rate at which any medium you wish will flow through the system by altering the pneumatic pressure. And that's what the box does. So there's a kind of gasket, and then the box will control airflow, which creates pressure, and you can calibrate how fast you want something to move. And then the medium is washed through the system. And then there's a flow cell, which is at the bottom of the plate, and so an inverted microscope can visualize what's going on. And on the bottom right, you can actually see this is what they actually look like. So you have an inlet well, then you have this great big long kind of serpentine of tubing that's all required to adjust flow rates, etc. And then you have two flow cells sit next to each other. So again, if you look at the diagram at the top right, you can see that pairs of inlet and outlet wells are kind of matched, and so you have two flow cells that sit together. So kind of 1 and 2 on the top left sit together. So to form a bacterial biofilm in this system is really simple, and you can do it in various ways actually, but our kind of basic standard protocol just outlined on the right here is to prime the plate. So you just basically fill media, so you get the flow cell so wet with whatever your medium is, and so you can just introduce the media and push it through. We then normally add a little bit more volume on the inlet well, so you have a gradient of liquid coming from inlet, from left to right, if you like, from inlet to outlet. And the reason we do that is that we then will introduce bugs from the outlet well. So we'll put some bacteria in what will eventually be the waste well, and then we'll apply a pneumatic pressure just to push them into the flow cell from right to left, so very brief. And then when the balance of the kind of solutions on either side is basically the same, there'll be kind of an equilibrium, and then the bugs will be entered into the flow cell, and then you can leave them to attach. It will really depend on your species for how long you do that, and it's generally not very long at all actually. And then you can start running a biofilm under whatever conditions you want. So you can then establish a flow rate from left to right for as fast or as slow as you want, for as long as you want. You can control temperature if you have one of the modules for the system that lets you have a temperature controlled stage. And then you can basically run your experiment as long as you like. You can remove the top gasket plate whenever you want, if you want to change media or add drugs or add dyes, if you want to stain anything or whatever. And then the plate you will then take off to look at by microscopy, or take it off to a confocal, or whatever you want. So that's in a nutshell the kind of very, very basic biofilm establishment protocol. So it's pretty simple, and I suppose from start to finish, a couple of hours, including the attachment incubation time, you'll start running your experiment, and then once it's running, you can generally leave it alone. So we would typically run a reasonably slow rate for 48 hours or so, and you can leave the system alone. Normally, you'll actually get about that amount of time of media, but you might need to top the media up once every 24 hours or so. But it's basically hands off once you've set it down. So that's a very, very basic overview of what it does. So we'll just go through a few examples of different things that we've used it for. So one of the very simple things you can do is you can just look to see how well different isolates will form a biofilm in the system. And so hopefully this kind of transmits reasonably to your screens. But here we have two strains. These are both Salmonellas. On the top 3 panels, you have a strain that's not a very good biofilm forming strain, and the bottom you have one that is a good biofilm former. And so the bottom one was actually isolated by an industrial partner as a produce contaminant, and the top one is a kind of lab strain that we know isn't a great biofilm former. And then you've introduced them into the system, and if you look in the left-hand column, this is after 2 hours, the middle is 12, and then the right of the match pictures is 24, and you can see at the top you get individual cells attached. You can probably hopefully see some of these, and even in the 24-hour one you start to see a bit of filamentation perhaps, but they're not really covering, they're not really doing anything, they're not making a biofilm, they're not really making any of the cement and matrix that you would associate with a biofilm. Whereas the strain at the bottom, which is a very good biofilm former, you can see at 2 hours is maybe not too dissimilar, so the initial attachment is the same. But this one can go and make a proper biofilm matrix, and so at 12 hours you can see that the flow cell is starting to be kind of covered, and 24 hours it's quite deep. And then on the right, this 48-hour panel is just a zoom out. So these left-hand guys are 40-time magnification, and the panel on the right is a times 10, I think, where you can see the poor and the good biofilm formers, the poor on the top flow cell, the good at the bottom, and you can see that you're getting kind of aggregates forming in big lumps of biofilm. So you can very easily just compare how well strains will biofilm if you wanted to compare isolate A, V, B, or if you've made a mutant or whatever. So that's quite easy. And then you can also take some of that kind of data and quantify it. So you can take, so what we did here is we took some replicates of these, and then across the flow cell we took boxes from the left, the middle and the right, and then we aggregated all that data. So for each one of these you've got a bunch of data points, and you can see that the poor biofilm forming strain after 24 hours is covering a very low percentage of the well. It gets a little bit better, And then in red, you can see the strong biofilm former is doing a good job relatively quickly. And that's something that the BioFlux's onboard software can do for you very easily, actually. So that's a very, very, very simple thing to do. Obviously, you can take this plate off to a confocal and look at it in more detail, which we'll talk about in a bit. But a very, very simple application is just to look at biofilm formation. Now, what we can also then do is, if we've established how well a strain will biofilm or will not biofilm, we can actually use this as a model to try and test the ability of anti-biofilm therapies to impact the ability to form a biofilm. So one of the things that we've been interested in is looking at ways to stop bugs from attaching to surfaces and stop them forming a biofilm. So just briefly, a little background to this. So one of our interests has been studying bacterial multi-drug resistance efflux pumps. So these are transport proteins that bacteria have in their membranes, and they have a very broad substrate specificity. So they will export antibiotics, dyes, disinfectants, detergents, lots of things. And so we've been interested in these for a long time for their impact on antibiotic resistance, but we've also found that they have a big role in the biology of the cell in other ways. So if you inactivate these, and this is genetic data where we've made specific mutants, we've knocked out various genes, then you see a big impact on biofilm formation. So the white bar is just an amount of biofilm formed by a wild type, and this data isn't BioFlux data. This is just the staining of biomass in a kind of microteacher tray. It's very simple assays, but we've done it in lots of different models, and broadly you see the same picture. And if you knock out any of these efflux transporters, then you see a reduction in biofilm formation. And this data is again all from Salmonella. And Salmonella has 9 multi-drug efflux systems. And if you get rid of any of them, then the amount of biofilm formed is significantly different. So that's fine, but you can't, in the real world, make genetic knockouts in strains. But we wondered if these targets could be inhibited as a way to block biofilm formation. And there are known inhibitors of these pump systems. And so we did a look to see whether these inhibitors can reduce biofilm formation. So can you chemically reduce the ability of a bug to form a biofilm? So there's no genetic knockouts here. So this is all data from a wild type strain. So it's the same type of data with, on the y-axis, you have the volume of biomass that's been formed. And now on the x-axis, you have the concentration of efflux inhibitors. And so on the top panel, you have a compound called PABM, which is one inhibitor. And you can see that as you go up the dose range of this inhibitor, the amount of biofilm formed drops in a kind of dose-respondent manner. And the bars that are kind of black are just to show that's where there was actually you begin to see a general growth inhibition effect of the inhibitor. So actually you can start reducing biofilm before you stop reducing growth. So this is a specific anti-biofilm effect. We see the same thing with CCCP, which is an inhibitor which works in a different way. And then a third one, chlorpromazine, again, works in a different way. So that all 3 of these inhibit efflux, but by different routes, and you see the same kind of pattern. So this suggested that these efflux inhibitors could be potentially useful tools to block biofilm formation. And this data, again, was with Salmonella, but we looked with various species. And this is some stuff from BioFlux. In this case, this is now Staph aureus. So in panel A, you have a strain of Staph, which is a strong biofilm former, and it's had no inhibition whatsoever. So you can see that this is after 16 hours. So this has formed a kind of fairly dense biofilm. And then B, C, and D are what happens in the presence of different inhibitors. And so actually you can see the amount of biofilm form drops off significantly, and you don't see anything near the amount of biofilm. And you get its initial attachment, but you don't get the maturation into a mature biofilm. And again, it's quite simple to look at these kind of impacts in the BioFlux and to see whether whether any new treatment will inhibit biofilm formation. Okay, so those are both kind of pretty simple. It's just using the phase contrast microscope to record what's happening. The nice thing about that is that you can do it all in real time. It's non-invasive. You can look repeatedly. You can do videos if you want to. You can do kind of real-time stuff. So we've done some stuff videoing the dynamics of biofilm formation, which I haven't shown actually. But you're not limited to light microscopy. So you can take your plate off and you can start using fluorescent dyes to look at what's happening in more detail. So we've also done some of this. So single cell viability under stress is something that we've been very interested in. So the idea here being that biofilms are heterogeneous groups of organisms. Oh, sorry, not heterogeneous groups of organisms. Often in the real world they are, but the cells within a biofilm are going to be in quite different physiological states, and that they will respond to drug stress probably in quite different ways. So one of the things we've done is to try and use the bioflux to look at this. So just again, kind of very, very brief overview of the approach is you grow your biofilm as you wish to the desired stage under your test conditions. So you can do this in whatever media you want, temperature, time, whatever. And then what we've tended to do is to kind of remove the waste and then flow through a carrier for our stressor. And when I say carrier here, generally we've used PBS. So we establish a biofilm in a media, growth media, and then we normally then will use PBS as the vehicle to deliver our stressor, which might be an antibiotic, it might be a biocide. You then add your stress, whatever that may be, chemical or whatever, it could be a physical stress. If you change the temperature, for example, and you do it for as long as you want, whatever your time point is. And then you can introduce any dyes you want. So obviously there are fluorescent dyes that will report on all sorts of aspects of biofilm biology. You can look for viability, you can look for matrix production, you can look for the presence of specific components within the biofilm. So extracellular DNA is quite common, or various specific polysaccharides. You can do whatever you like. In this case, what we're going to talk about is using two dyes here called cyto, one of the cyto dyes, which is a cell viability stain, and propidium iodide. I'll explain how they work in a second. So you introduce your fluorescent dyes, you incubate them, wash off any excess dye that's unbound, and then you can take it to the confocal and away you go, and you can visualize what's happened. So again, it's a pretty simple kind of approach. There's a bit of tinkering to get the incubation conditions, etc., correct, but we found it to be relatively straightforward and simple. So this, just to show you, this is the kind of data that you can get, or some of our data. I'll just explain what we're looking at here. So these are some biofilms which have been stained with two dyes, a green one, Cyto 9, and Cyto 9 will basically light up all cells and it will paint them green. And then propidium iodide, which is a red dye, and that will only go into dead cells. So basically propidium iodide, normally a live cell will exclude it from inside the cell and they won't be stained. So essentially what you see is that live cells will look green. So in F, you see this is a kind of control where you basically just got a wild type biofilm with no stress. And you see pretty much everything is alive. There's a few maybe red cells at the bottom in the background who have probably not got much access to nutrients for a while. E is the kind of kill control. So this is a biofilm that's been exposed to 70% ethanol. And you can see that basically everybody's dead, as you would expect. And these pictures at the bottom are slices. So these are kind of 2D slices through the biofilm that the confocals picked. So you see some stuff looks in kind of quite sharp contrast, some stuff looks fuzzy because it's probably a layer below or above. And then B, C, and D are biofilms that have been exposed to different oxidizing stresses. So again, B is hydrogen peroxide, C, peracetic acid, and D, sodium hypochlorite. And what you can see is you get these different patterns of survival. So you see different degrees of killing. You see some green cells in all of them, but the degree and the location of them differs a bit. And you also get some more or less kind of destruction of the biofilm with the different treatments. So you get less actual coverage of the flow cells. And then what you can also do is, as you can see in A, is you can take a Z-stack. So you can actually kind of overlay this information together. And that's quite useful because you can get a 3D idea for what's happening in the biofilm, which might be useful if you're interested in penetration. So some drugs will struggle to penetrate deep layers of biofilms more or less than others. And actually one of the things that we found here, which was interesting, is if you look at A here, you can see that this is a biofilm that's largely okay, you can see green, but you get these kind of islands of killing. So you get these kind of red blobs, and they actually, when you look, they actually will go all the way down through the biofilm. So actually the depth in the Z stack, if you like, in the Z dimension of the biofilm doesn't seem to be that important, or the Y dimension doesn't seem to be that important. So survival or not, it seems to be where you are. Is it you get like an island of kind of killing or not? And we've got some ideas about what might be prompting that, which we're following through. But this is the kind of stuff that you can do. So you can start looking at kind of viability. You can kind of get into interesting impacts of different treatments. You can look at different strains to see if one is more or less hardy than another, etc. And you can look at different ages. So this first data, this is a 24-hour biofilm, and a 48-hour old biofilm is you can see you get these bigger bits of biomass in areas, and some of those big mountains, if you like, most stuff is alive, and so there is some evidence there that depth of the biofilm is protective, is important. So you can start asking these kind of questions, and you can generate a lot of information from these images. And so what you can do, it depends on the bioflux system you have, but the number of flow cells that you can run concurrently varies. So we typically run 12 at a time for various reasons, and so you get a reasonable number of different isolates or different exposure conditions, and so you can kind of compare what's what. But you can do various bits of analysis on this stuff. So for example, one of the things that we were interested in, one question we're interested in asking is, are the surviving cells where you're seeing stress randomly distributed or not? So the kind of, this is just one picture here at the bottom, and you can kind of see green cells dotted throughout, and by eye you kind of look and see, okay, these guys look like they're spread throughout the biofilm. So this is just one slice. Again, you can see some of it's in sharper focus than other just because of the depth of the biofilm. But you can actually ask a question of that. So so we took these images and we put them into image J, and there's some simple plugins for that, which will actually you can basically look for where are all the red particles, where are all the green particles, and then you can ask the question, are the green particles, is their distribution random or are they clustered? So we had the hypothesis that these were actually randomly spread because they looked by eye that there wasn't any kind of clustering. And actually, if you test that, and this little graph shows the test, so basically the black line shows you what you would see if they were completely randomly clustered, and actually the red line is actually the data, and actually they are clustering, which tells us that when you then kind of look at it knowing that, you can see actually maybe you're more likely to see a green particle closer to another green particle than completely randomly spread. So maybe there is actually some kind of, these guys might be progeny of ones that have survived, we're not sure. But you can kind of use the information that you can get from the system to take it forward to ask more sophisticated questions. So looking at biocide killing in this case has been quite useful for us, and we've got quite a lot of information about the dynamics of responses. One of the other things that we've been interested in is looking at expression of genes within biofilms. So when you can look at viability, and that's fine, but that doesn't still tell you the mechanism for why some cells might be surviving and might be viable. So again, there are all sorts of fluorescent reporters that have been used to look at gene expression or the presence or absence of particular proteins or molecules in biofilms. Anything that you can see through a piece of glass is compatible with use in the bioflux. It doesn't really matter. So we've done a little bit of this where, again, you grow your biofilms just as you would or however you want. You add whatever stress you're interested in, and then if you've put a fluorescent reporter into the biofilm, so we've done this with strains that are hooked up to, or have strains which have genes that we are interested in that are linked to GFP, so that they will produce GFP when they're turned on, and then you can look at their expression over time. And again, you can do this whenever you want. You can do it how often you want, and you can do it plus stress A, B, C, or whatever. And you can also multiplex things together. So you can actually look, you can say, well, we can capture viability plus gene expression together. It depends on your confocal, but it's quite common and standard for most decent confocal microscopes. We'll be able to capture 3 colors fairly happily. So you could quite easily look at viability and gene expression together, or you could look at expression of 2 genes plus presence of a protein that you're interested in or whatever. So again, it's a kind of nice application. So this is some information showing expression of one of the genes we're interested in. On the left here, we've got just these are colonies going on plates, and in this case, the gene ACRB, which is one of those efflux pumps we're interested in, has been hooked up to a GFP reporter, and you can see that at 24 hours, this colony is producing most of its ACLB at the edge, so where the cells are growing. And then at 48 hours, the kind of colonies mature and start to make these kind of wrinkly biofilms. You can see the green fluorescence is really only at the edge. So we know that there's a kind of growth-dependent expression of these things, which is interesting. And we also then had a look in a biofilm. So this data probably doesn't show up as nicely, actually, because it's a bit zoomed out. But at the top of the right-hand panels, you have a biofilm that's under no stress, and you can see that basically there's very little, there's not much green going on, so these green foci of expression. And then if you add, in this case, peracetic acid, you kind of see these flares, these little foci. There's a lot more red because everything's beginning to die. So again, these have been stained with the PI. But you see these little flares, these foci, but across the whole biofilm, most of it doesn't respond, but you see these kind of little subpositions that do. So we get some information about responses, and again, you can zoom right in and see what's going on in these particular parts of the biofilm, and whether it's the bottom, the top, whatever. So you can look at gene expression. The other thing that we've kind of had a play with is using the biofilm to look at, the bioflux to look at how biofilms can adapt to stress. So classically in antibiotics, in the antibiotic world, if you're looking for how bugs can become resistant to a stress, then what you do is you grow a big population, you apply a big stress, and then you just look for anybody that can survive. And so you'll grow a big broth of bugs, you'll throw in a big concentration of an antibiotic, for example, you will then plate them on agar, and you will look for anybody that can grow, and you'll look for then mutants that have got an ability to survive, and then you can try and find out why. So what's different, what's their mechanism? So that's fine, but we've been interested in, and we are interested in the moment, in how do biofilms adapt to stress. So we've used the bioflux to have a go at this. So basically, kind of very simply, we asked the question, can you actually pull out mutants? Can you basically form a biofilm, and then can you use the system to shed cells from it and recover them? And we're able to do that. So actually, we can grow a biofilm, and you can grow it up to whatever, 24, 48 hours, a normal biofilm as you would, and then you can kind of clear out the waste from the system, and then you can add a stress. In this case, this again is a very high concentration of a disinfectant, peracetic acid, so a biocide. And what we did is we actually showed with control wells that actually what you can do is you can recover a lot of cells from a biofilm. So you can get up to about 10 to the 7 colony forming units. So you can actually by just applying a high rate of pressure, so you don't need we thought we might need to have to add bits of SDS and things to try and remove cells gently from the substrate, whatever. But actually just applying a high force, you can recover large numbers of viable cells from the biofilm. And doing that, we did the experiment under stress, and we found that 2 cells we could recover that had survived from the biofilm. And these guys were actually mutants that were more resistant to peracetic acid. And they were also very resistant to beta-lactam antibiotics, which was interesting. So this kind of little table on the right just shows the concentration of various antibiotics needed to inhibit their growth, and 828 is the parent strain, the kind of control arm, which gives you various values. And then these 2 mutants had an identical profile, so it's probably actually the same thing that's had a baby. So you've had a mutant that's then generated a daughter cell, and it's very highly resistant to beta-lactam antibiotics, and we still haven't figured out the mechanism for that yet. But the nice thing is that we are able to recover quite decent numbers of cells from a bioflux, and in theory what you can do is you can then take the cells from a biofilm and then feed them to another flow cell and iterate and kind of use it as a kind of system to adapt and evolve strains over time in this context, although it wouldn't be the highest throughput way of doing that. Okay, so the other things that we're kind of interested in following further are really getting into the dynamics of this survival within a biofilm more. We know there are clear subpopulations in the biocide-exposed biofilms or antibiotic-exposed biofilms. So are these mutants, or is this just the physiological variants that you see within a biofilm? So we're going to start asking questions about what's the matrix composition at different parts of the biofilm, how metabolically active are these cells, and again, you can use fluorescent reporter dyes to report on that kind of information and overlay a viability with it. And what we're doing at the moment is a lot of work to look at, can biofilms evolve resistance, and how do they do it over time? and looking at things in the bioflux when we've got these evolved populations. So I've got a few kind of conclusions and then a few kind of maybe hints and tips and things before we'll kind of see if there's any specific things people want to ask. So we've had a bioflux for a fair while. We found it to be versatile, and as hopefully have shown, we've used it for various things. And the ability to study biofilms in situ is quite useful for us and has proved quite powerful. You can look at various media. Incubation conditions can be easily modified. You can add interventions, you can add drugs, you can change nutrient sources, whatever. You can add multiple species together. It's very easy to take the plate to a confocal microscope, so this single cell analysis is very useful for us. You can look at gene expression. You can also collect surviving cells from a bioflux, which is quite cool. And looking at the host pathogen interactions is something that I haven't touched on today, but you can grow human cell lines in biofluxes. In fact, that's what they're probably mainly used for, I would guess. And then you can introduce bacteria onto those and look at host pathogen interactions. I mean, there are limitations. The bioflux is large. The substrates that the flow cells are made of are kind of limiting to some extent. Incubation conditions are not necessarily suitable for all organisms. It's not the most high throughput system in the world, so there are limitations. So what we tend to do is to do our kind of, if we're do some of our experimental evolution experiments, we tend to do those in kind of quite big parallel ways in the laboratory, and then when we find strains that have evolved or adapted or mutants that we think are interesting, we will then look at those in the bioflux. And so we use it as a tool to kind of look at in more detail the dynamics of what happened to our kind of our populations that have been exposed and evolved. Okay, so those conclusions. A couple of maybe kind of tips and tricks about the stuff we've done. So attachment time can vary between species. If you want to form a biofilm, some people have found they need to leave cells in there a bit longer. We've actually found that most bugs will attach pretty rapidly, but some strains are very, very good biofilm formers. Some Acinetobacters we've used are very aggressive and form very strong biofilms very quickly, and that can be a problem in that, as I said at the beginning, you introduce your bugs, or we do, from the outlet well and let them sit in the flow cell. If you let them get upstream too much, they can actually colonize that very fine serpentine, and actually they can block that if you're not careful. And actually quite aggressive biofilm forming strains can actually start growing up that. There are ways you can get rid of that. So we had to kind of add pulses of slightly higher flow for short periods of time to kind of keep that clear for some of the Acinetobacter work we did. And again, probably one size doesn't fit all. To try and get a biofilm that's going to suit your needs and whatever you're trying to model will depend on what your biological interest is. We've been particularly interested in looking at what happens in kind of hospital water systems, where this is a nice model to do it, because you can grow things actually in water with relevant strains, and you can do it with flow rates that are sensible. You can add disinfectants at time points that make sense, but it really depends on what your particular model is, as to how you should run the system. So that's kind of pretty much all I wanted to say. Hopefully that's given an overview that will be useful to some people of all the bits and pieces we've done. I should just say thank you to the people who've kind of actually run most of those experiments. So kind of Steffi Boff, Kerry Guest, Marta Arch, and Alex were all students who've used the system in my lab in the past and kind of published some of the stuff that I alluded to. So yeah, I think that's pretty much what I wanted to say and give an overview of how we found this useful. And if there are any questions that people have, I'll do my best to try and kind of answer them. I know I didn't go into any kind of detail with any protocols, and we can kind of give more details about how exactly we do things off, if people want, or Fluxion can help, I'm sure. But I wanted to just give a kind of overview of how we found the system to be useful. So that's me. I'll shut up now in case.
Thank you, Dr. Weber, and congratulations on a remarkable body of work. Everyone's encouraged to put questions into the question box, and I'll start with mine, which I think is probably the most important. Did you ever meet Ozzy Osbourne when you were in Birmingham?
No, I didn't. I have spent time very near where he was from, but no, I never met Ozzy myself. I think he was too busy in LA by that point, actually.
So going to a 50,000-foot picture, where do you think the trend is for antibiotics in the next 5 to 10 years? Or in the past 5 years, a lot of people have been going back to older antibiotics like penicillins. We've had a couple new antibiotic classes put in, like the cubicin, but not much. Or do you think that we'll just be doing, say, modifications of existing antibiotics like fluoroquinones, or that we'll actually come up with new, completely new antibiotics?
So I think we'll probably my guess in the short term is what we will see is you will see modifications, derivatives of current classes, and that's really what has happened in the last 50 years. For most of our new antibiotics have been modifications, derivatives of classes, and there'll be still some mileage there. I think that what you will see is improved diagnostics. So there will be tests that will kind of be able to tell you more quickly, and some of those are microfluidic based actually, what the infective organism is and what it's sensitive or not to. So we'll be able to use our current drugs better. Truly new classes is probably further away. So there are people looking, and a lot of that work is going back to natural products again. So looking back in, most of our antibiotics come from produced by Streptomyces. And people are looking in a much wider range of Streptomyces species, and there are a lot of potential antibiotics that they might be making. You can see the biosynthetic gene clusters on their genome. So looking for new drugs, going back to natural product discovery, I think will yield new drugs, but whether they will be broad spectrum kill everything drugs as we've had in the past, I suspect maybe not. And we might move to a paradigm where if you have a diagnostic test that can say you have an infection with a Klebsiella, for example, in a reasonable period of time, then actually there's a window for antibiotics that will not cover everything to be useful. And you can say, okay, we can look for drugs that will deal with 2 or 3 species, and we don't have to have things that will necessarily kill everything. So I think that will be hopefully progress in various areas, but in the very short term, I think the pipeline of what's coming is derivatives of current stuff or combinations with inhibitors.
Well, great. And a more technical question from me, and then we have a couple questions that came in. When you go to treat with antibiotics, how developed do you, and you mentioned this before, that there's not one size fits all. And many of us know that if you have an aggressive biofilm former in the bioflux, that it will plug eventually to the point where the biofilm's completely blocked. Do you have a rule of thumb for when you introduce these antibiotics into the system?
Yeah, so actually we've managed, we normally can stop it plugging by, just to address that point, we've just doing a kind of short pulse of high flow, like literally 10 seconds, 5-10 seconds, just to kind of increase high flow, short period of time. We'll normally kind of get rid of the we'll get rid of the plug. Now, when we add our drug is an interesting question. So kind of at what point should you say that you want to kind of say your biofilm is ready to be tested, if you like? So that's a good question. And again, I'm not sure there's a right, it really depends on what you're trying to model. So for us, it depends on the species. A 48-hour biofilm for Salmonella would be a fairly mature biofilm, if that makes sense. A Acinetobacter, they will biofilm some of the strains very aggressively. Kind of 12 hours, they kind of look pretty mature, but there will be differences between a very old biofilm and a very young biofilm. So I guess it kind of depends what you're trying to model when you throw your drug. But if you attack a biofilm early, you will kill more of it than if you do it late, if that makes sense.
Sure. I've got a question here about, and getting a little bit into the weeds here, about how you collect the survivors when you go to do, I guess this applies to the bioflux specifically. So you've developed the biofilm, you've treated with the antibiotic, and you mentioned a pulsing. Do you have a rule of thumb for that, or it's, again, not a one-size-fits-all, but depending on the bug and how adhesive it is, how long you take to pulse the material out of the channel in order to plate it out?
Okay. So what we did with that to try and recover cells from the biofilm is that the first thing we wanted to do is just increase. So most of the biofilms we run at a relatively low flow rate, so like 0.3 dyne or whatever, so quite low. And then you will see that you get some bugs that will then grow planktonically in the kind of in the outlet well, because it's just media, right? So it's just, and you can see that's fine. So what we tend to do is we kind of clear out the waste. So basically empty that out, and then what we tend to do is just have a period of a kind of slightly higher flow, so maybe a dyne, something like that, to just try and really wash out kind of loosely bound on or cells that aren't part of the biofilm, and let that run for 20 minutes, something like that. And again, then kind of clear out anything that's come through in that kind of period. And then what we tend to do is then to remove cells that we actually want to get off. We add quite a high dyne, so we kind of go up to 10, 20 dyne, so quite high flow rate. And again, if you let it run like that for 10 minutes, 20 minutes, then you start to get those numbers of cells. And again, if you do that in just a wild type biofilm with no stress, you get your kind of, we managed to collect about 10 to the 7 cells. So actually what we then do is collect all those cells, which will include big lumps and bumps. We then will kind of take those off and vortex them to try and break cells down to individuality. And then we can kind of plate them out and look for, in the stress arm, we actually get very small numbers of survivors. And yeah, does that answer the question?
Oh, absolutely. And to keep moving along, I guess we'll cut this off at a certain point, but we did actually get quite a few questions, which is quite exciting.
Okay, cool. I can't see those, so yeah, if you just read them out, then that's cool.
So someone asked just briefly, do you think the bioflux could be used for microbiome research?
It could. So I guess the one difficulty there would be the complexity of the community, which is the difficulty with doing microbiome research when you come out of a real microbiome environment, is trying to recapitulate the real diversity of the community, right? So that's the problem that the microbiome people. So our institute, the Quadrium Institute, is going to have a huge microbiome focus in the next few years, and that's one of our kind of big drivers, and that's largely gut microbiome focused. And there are people interested in using the bioflux to look at interactions between communities, and that's definitely possible. So looking at kind of communities is absolutely fine, and where people beginning to understand how you can keep 4 or 5 species together happily, and trying to make laboratory models of what you see, in our case, in the gut. It is really difficult though to recapitulate a proper gut environment in a microfluidic platform. What I think you will be able to do is say, okay, if you'll be able to grow, say, we're going to grow bifidobacteria, lactobacteria, kind of ruminococcus or whatever. We'll probably be able to work out ways of doing that. And I think we've grown like 3 or 4 species together and managed to get them to stably coexist. We've done that. And then you can add a stress and you can see, who does it affect? Does the community fall to bits if one of those component members is not there, for example? So to kind of take a reductionist approach and ask some of those key questions, yes. And the advantage you have is that you can, obviously, if you've got ability to label your different members differently with fluorescent dyes or whatever, you can actually see what's happening in real time. But there are limits, and you're not going to recapitulate a human gut in a kind of tiny flow cell, I guess. But you can start to kind of probe those interactions. And the way that it's being used, or people are interested in using it here, is taking data from kind of large metagenomic studies and looking at patient cohorts and saying patients that tend to have a drop off in bug X seem to have an over amount of bug Y. Does bug Y inhibit bug X? And you can start actually throwing those 2 guys together in competition and look in the bioflux and look for that kind of those interactions.
That's quite interesting. We've had actually 2 questions here that relate to where current antibiotics are, and either looking at things that maybe target, say, quorum sensing that you touched on, or that development of antivirulence factor drugs, which I guess are sort of in that same ballpark, whether they be adhesion or efflux pumps or other types of mechanisms around that. Do you have any thoughts on that?
Yeah, so our goal is to stop infection making you ill. And so actually, if you can neutralize a bug, if you can have a drug that stops it causing disease, then you don't necessarily need to kill it. Your immune system is good at killing bugs. So actually looking for virulence blockers is potentially quite an interesting approach. It would kind of probably depend on the context and what you want to do. But if you had something that really very, very robustly neutralized the bug or stopped it from causing damage, then that potentially could clinically be quite useful. And obviously quite a lot of our antibiotics at the moment don't actually kill bugs, they stop them growing. And if you limit the ability of a bug to elaborate a virulence factor, then maybe it cannot survive survive and grow in its host anyway, in its context, in the niche that it's trying to colonize. So there are people looking at that, and there are some interesting proof of principle. Quorum sensing inhibitors have been looked at for ways to block biofilm. I'm not sure there's anything that's close to being clinically that useful, but as an approach, it's interesting, and obviously it's not something that's been overmined in the past, because when we looked for antibiotics, we looked for things that killed bugs or inhibited their growth, not for things that modulate their behavior. So it's in theory untapped space, and there might be a lot of mileage in there. So I think it's quite an interesting idea, and it's something that we should definitely consider and look at.
That's quite interesting. And getting back into the weeds a little bit, when you incubate, so the two probes you primarily talked about were Cyto9 and PI. And in my hands, I found that, occasionally that the combination, if you're doing a kinetic, a time-lapse image plus the fluorescence and the probes themselves could be inhibitory to the biofilms. Do you have a preference to maybe putting the dyes in ahead of schedule along with your antibiotics, or doing that afterwards? And one of the things I found doing this afterwards, especially with something like a staph, like a big comet of staph, is that it forms this extra polysaccharide shell around it. And just as it's great at repelling antibiotics, it's also quite great at repelling, say, PI and Cyto9 from penetrating in.
Right, okay. Yeah. So we did most of our stuff with gram-negative. We didn't really, I mean, you have to play with the concentrations of the dyes, actually. That's definitely true to get the right permeation. And we have done it both ways. We have done it where the dyes have been basically kind of drip fed in with the stress to try and look at short-term responses, rather than saying that we're going to now attack you and then we'll stain you and see what happened after the event. So we have done it both ways, and it works actually okay. You can drip it through. The staining happens fairly quickly. So in our hands, at least, they stain within a couple of minutes fairly well. The data I think I showed you was probably stuff that had actually been kind of stained and let to sit and then washed out, and we normally would leave that for about kind of 20 minutes or so. But actually, we have done it where you kind of feed them in. It depends on the dye. And some of the fluorescent dyes are better at permeating cells than others. Some are actually staining bits of the cell that are more accessible than others. So again, it depends. And some bugs probably make more slimy matrix again than others. So some would be harder to deal with. But there are so many fluorescent reporters that I think you would probably always find something that would be useful for whatever the specific question you're trying to ask is.
Sure. And then one more big picture question before we finish up. And, we talked about this a little bit yesterday, but to rehash the question about finishing the full course of antibiotics versus, you feel better, does your body take over and finish the job off, either empirically or based off of actual clinical data? What are your thoughts on this?
Okay, so that's a really interesting question, and it's been the topic of quite a lot of debate this year, actually. So there was a very provocative piece in, I think it was in the Lancet or the BMJ, which essentially said that, or the way it was reported was actually doesn't really reflect the data actually, but the reporting was, when you feel better, stop taking your drugs. Okay, so like that's okay, that'll do. And actually the data in the paper didn't really say that, it didn't support that. What there is though is there's been a move in the last few years for shorter dosing regimes, but maybe shorter, sharper dosing. So actually it's surprising how little we know about how you should dose an antibiotic. So most of our antibiotics are very old. Their clinical trials were done a long time ago. No one's going to pay for the clinical trials now because they're all out of patent to actually establish the pharmacokinetics as well as you might. But where there is evidence generally, and this again is very, very case by case and drug by drug and bug by bug, so you shouldn't take a kind of one size fits all, but I would say generally the evidence is pointing that actually shorter courses, but with maybe higher doses to start, higher doses work as well as long ones. And so if you kind of, historically we gave people kind of drugs for a week, so okay, take it for a week for maybe a kind of an ear infection or whatever. Probably the evidence now suggests that you could have a higher dose for 2 or 3 days And actually that's going to do the job, and it actually has less risk of selecting for resistance because your kind of window with a lower concentration for a longer period is lesser. But it does depend very much on drug by drug. And I think one of the dangers about that kind of paper earlier this year was then kind of the implication that, okay, the minute you start feeling a bit better, stop taking your drugs, which actually, if the infection hasn't actually been properly cured, got rid of, you will just get a bounce back. And actually that can be dangerous because you have more chance for things to become resistant. And TB has been the classic example of that, is that the drug therapy takes a very long time. People don't like to take it. They start feeling better and stop taking it, but then actually they relapse and you can select resistance over time. So I guess the general rule of thumb seems to be the opinion is moving to shorter, sharper courses of drugs that are equally effective, but it's very much case by case. And obviously you should follow whatever the doctor's advice is on any particular course.
Right. Well, I really appreciate this again. Congratulations on a great body of work, and we look forward to even more exciting stuff down the pipeline. There were a couple other questions that we'll answer offline here. And I guess in the U.S., good morning. In the U.K., good evening. And in Asia, for the few of you who are over there, go back to bed. Thank you.
No worries. Thank you.